All right, welcome to your GCSE Biology revision. If you're tackling section 4.1.2 on cell division, then you've come to exactly the right place. Let's get right into it. I mean, think about it. How on earth do you go from being just one tiny cell to, well, you, with trillions of cells all doing different jobs? The answer is this amazing process, and really getting your head around it is going to be key for your exams. So, here's our game plan. We're going to follow the entire story of a cell. We'll start with the instructions, the blueprint if you like. Then we'll see how it actually copies itself. After that, we'll look at these incredible supercells and what they can do in medicine. And finally, we'll get into the big debate around them. Ready? Let's go. Okay, first things first. Before a cell can even think about dividing, it needs a set of instructions to copy, right? And that's where chromosomes come in. These are the absolute master plans for everything. Okay, so here is your official exam-ready definition. Super important. Inside the nucleus of a cell, you find these structures called chromosomes, and they're basically long, coiled-up molecules of DNA. And a key thing to remember, and the exam boards love this detail, is that in our body cells, they're normally found in pairs. Yeah, to make this really, really simple, just picture a chromosome as a complete instruction manual for building you. It's that straightforward. And your body has 23 pairs of these manuals tucked away in almost every single one of your cells, telling each part exactly what to do. And this just breaks it down perfectly. It shows you how it all fits together. So the chromosome is the whole manual, the whole book. The DNA, that's like the individual pages inside the book. And a gene, well, a gene is just a small section of DNA on one of those pages, like a single recipe or a single instruction. Right, so we've got the instruction manuals, but how does a cell actually go about copying itself and all those instructions? This is where we dive into the cell cycle and a really crucial part of it called mitosis. So the cell cycle is a really precise 4-step journey. Think of it like a little life story. First up, the cell just grows. It gets bigger and it makes more of all its internal bits and pieces, like mitochondria and ribosomes. Then step 2, and this is absolutely vital, it copies its DNA. It makes a perfect duplicate of every single chromosome. Step 3, mitosis itself kicks in. The 2 identical sets of chromosomes get pulled to opposite ends of the cell, and the nucleus splits in 2. And finally, step 4, the rest of the cell, the cytoplasm and the membrane, divides right down the middle, and boom, you've got 2 brand new genetically identical daughter cells. So, what's the big takeaway here? It's all about the result. You start with one parent cell, and it divides to create 2 daughter cells that are perfect clones. They're genetically identical to the original and to each other. That is the whole point of mitosis. But why should you care? Why does any of this matter? Well, it's pretty simple, really. This process, mitosis, is literally how you grew from a tiny embryo into the person you are today. It's also how your body heals a cut or replaces worn-out cells. It is happening inside you constantly, right this second. Okay, so most cells are pretty straightforward. They divide to make more of the same type of cell. But what if there was a cell that could become anything? Well, there is. Let's talk about the superheroes of the biological world, stem cells. And here's the official definition you'll want to have locked in. A stem cell is undifferentiated. That word is the key. It just means it hasn't decided what it wants to be when it grows up yet. It hasn't specialized. So it has this incredible potential to divide and then change or differentiate into all sorts of other cell types. Okay, so there are different types, and this is a really important distinction to make. You've got embryonic stem cells, which, as the name suggests, come from very early embryos. These guys are the most powerful. They can basically turn into any type of cell in the human body. Then you've got adult stem cells. We find these in certain places like our bone marrow. They're still incredibly useful, but they're a bit more limited. They can only turn into a certain range of cells, like different types of blood cells, for instance. Now, for your exams, you need to know the key places we find these. You've got human embryos and adult bone marrow in animals. And don't forget plants. In plants, you find them in what's called meristem tissue. This is why you can take a cutting from a plant, stick it some soil, and grow a whole new one. Because those meristem cells can differentiate into roots, stems, leaves, the whole shebang. So this ability to turn into different cells isn't just a cool party trick for biologists. It has absolutely massive, potentially revolutionary implications for medicine. Just think about the possibilities here. Scientists are working on using stem cells to treat conditions that we currently have no cure for. Imagine being able to grow new nerve cells to help someone who is paralyzed walk again, or growing new insulin-producing cells for a person with diabetes. This isn't science fiction. It's the incredible promise of stem cell technology. But, as with many big scientific breakthroughs, with great power comes, well, a really big debate. And listen up, because this is a super important part of the topic. This is where examiners love to put those big, juicy 5 or 6 mark questions. They don't just want a list of good things. They want you to evaluate. And that means you've got to look at the whole picture, the benefits, the risks, and all the controversy. Think of this slide as your secret weapon, your perfect answer plan for one of those evaluation questions. The way to get top marks is to build a balanced argument. So you pick a point from the benefits column, and then you counter it with a point from the risks and objections column. For example, you could say, stem cells offer amazing potential to treat diseases like paralysis. But then you immediately follow up with, however, there are serious ethical objections to using embryos to get these cells. See, you're showing the examiner you can think critically and weigh up both sides. That is what will get you those top marks. And that's where we land, right at the point where incredible science meets some really tough societal questions. The journey of a cell, from a simple division into two, all the way to the mind-blowing potential of stem cells, it opens up a world of possibilities. But it also forces us to ask some really difficult questions. So for your revision, remember that whole journey and make sure you're ready to talk about both the amazing promise and the tricky problems. You've got this. Good luck.